Welcome again. In this section, we'll be talking about the components and origins of axial skeleton development. I'm your host, Sannan Rehman. So, a brief introduction about this section. We'll talk about the various components of the axial skeleton, how the embryological development of axial skeleton takes place, and the process of ossification, the types and cells involved in the process, and how does the process of ossification actually occurs. Now let's get into it. First of all, the introduction. The skeleton is divided into two subgroups. The first subgroup or the first part of the skeleton is the axial skeleton. Axial skeleton consists of the skull, the vertebral column, the ribs, and the sternum. Now, the second component of the skeleton is the appendicular skeleton. In appendicular skeleton, we have the upper limb, the shoulder girdle, the lower limb, and the pelvic girdle. So, for the ease, for the convenience, and also from functional point of view, the skeleton is divided into two parts: the appendicular skeleton for the appendages, and the axial skeleton, which is the central skeleton in the body. We'll be talking about the development of this central skeleton, which has the four basic elements, which are skull, vertebral column, ribs, and sternum. So, embryological origins. Paraxial, lateral plate, and mesoderm and neural crest cells are the sources from where the axial skeleton originates. They unite to form somites and somatomeres. The ventromedial part, here in this figure, this is the ventromedial part. You must have a clear idea of the dorsal and the ventral surface. If I demonstrate it on my hand, this is the ventral surface and this is the dorsal surface. So keep that in mind while constructing a picture of embryo in your mind, or while looking at this picture I have attached in this slide, so that you have a clear idea about how the development is taking place. So now where my mouse cursor is, this is the ventromedial part of the somite. This part is known as the sclerotome. The sclerotome forms the mesenchyme or the embryonic connective tissue, which gives rise to the fibroblasts, the chondroblasts, and the osteoblasts. Okay, the fibroblasts lay down fibers in the extracellular matrix and help in its formation. The chondroblasts are the cartilage forming cells, and osteoblasts are the bone forming cells. Now, as a general rule, whenever blast comes with a cell type, it is usually a maker cell, a cell that helps to construct something. However, there are no laws in medicine, as I've told you before. So it is not necessary that whenever blast acts as comes in the ending, it may be a maker cell or a constructor cell. So now we have the dorsolateral part. This is the dorsolateral part where my mouse cursor is at the moment. This is the part of the somite which forms the dermomyotome, the dermatome and the myotome, which is responsible for formation of muscle as well as the skin. So mesenchyme of the lateral plate mesoderm forms the bones of pelvis and shoulder girdles, limbs, and sternum. So pelvis, shoulder girdles, and limbs, these are the parts of, yes, appendicular skeleton. However, sternum, the only part of axial skeleton that is formed from the mesenchyme of the lateral plate mesoderm, is sternum. So mesenchyme of the neural crest cells of the head help in face and skull bones formation. Occipital somites and somatomeres help in cranial vault and base of skull formation. I know it's a mouthful, I know it's a little bit difficult, but it has to be memorized the way it is. There are no easy ways, however, you must repeat it again and again until you're able to memorize it. So now let's take a look on ossification or osteogenesis. What is that? This is the process of laying down of the new bone. It involves two types of cells, the osteoblasts and the osteoclasts. Now, the osteoblasts are the bone forming cells. These are the cells which are responsible for formation of the bone. As you can see in the figure, they are single or mononucleated, well formed, well defined cells, in contrast to osteoclasts, which are multinucleated and giant cells. They are responsible for lysis or dissolution or destruction of the bone. The action of both osteoblasts and osteoclasts is necessary for the formation of bone. We have two processes through which ossification takes place. The first is intramembranous ossification and the second is endochondral ossification. Now let's take a look on these processes of ossification in a little detail. Intramembranous ossification. So it occurs more commonly in flat bones. The mesenchyme in the dermis differentiates directly into the bone so that a model of the bone has already been formed. Intramembranous ossification follows 4 steps. Mesenchymal cells, first of all, they group into clusters. These clusters differentiate into osteoblasts and ossification centers are formed. So initially we had the mesenchyme. The mesenchymal cells formed a cluster. This cluster differentiated into osteoblasts, the bone forming cells, and an ossification center, that is a center from where the formation of bone begins, is formed. Now, the secreted osteoid traps osteoblasts. This ossification center secretes osteoid. Osteoid is an extracellular matrix which is rich in bone formation material, which then becomes osteocyte. Osteoblasts, which are trapped in osteocytes in osteoid, now become mature bone cells which are known as osteocytes. Trabecular matrix in periosteum is formed, and lastly, compact bone develops superficial to the trabecular bone, and the crowded blood vessels condense into the red bone marrow. In the next slide, we'll take a look on the following process pictorially. This is the first process in which the mesenchyme arranges itself. The mesenchymal cells form osteoblasts. They tend to secrete osteoid, and an ossification center is formed. In the second process, in the second step, these osteoblasts which were trapped within the osteoid becomes osteocyte. Osteocyte is a mature bone cell. In the third step, the mesenchyme forms the periosteum, the outer lining, and the trabeculae. The blood vessels enter the trabeculae. In the last section, the fourth step, the fibrous periosteum is formed. The osteoblasts now form compact bone outside the trabeculated bone. The spongy bone or the trabeculated bone is inside, and the compact blood vessels now form the red bone marrow. Now we move on to endochondral ossification. In endochondral ossification, bone forms by replacing the hyaline cartilage. There are 5 steps in this process. The mesenchymal cells differentiate into chondrocytes that produce a cartilage model of the future bony skeleton. So first of all, we have the chondrocytes. The chondrocytes form a cartilage model of the future bone that has to be replaced and formed into a true bone. In the next step, the blood vessels on the edge of the cartilage model begin to deposit the bony collar. This is the bony collar being deposited. In the next step, the capillaries tend to penetrate the cartilage and deposit bone inside a cartilage model. This is the cartilage model. Spongy bone is being formed within, forming the primary ossification center. This forms the first or the primary ossification center. In the next step, the cartilage and chondrocytes continue to grow at the ends. At the ends, the cartilage and chondrocytes continue to grow, while the medullary cavity, this is the medullary cavity, expands and remodels. Secondary ossification centers are formed after birth. Hyaline cartilage remains at the epiphyseal growth plate. This is the hyaline cartilage at the epiphyseal growth plate and at the joint surface as the articular cartilage. In this way, a cartilaginous model is converted into an adult bone model. This was all about the introduction, the various forms of ossification in the human body. I hope you found it interesting and you are excited to learn more about this. For that, you have to keep watching sqadia.com. Thank you.